Hello my dear aspirants, I hope you all are doing well and I hope your preparations are going well for the SLET exam. So we have only a limited time left for the Northeast SLET exam and the exam is going to be held on 23rd March 2025. I hope you're in this last few days what all strategies that you have to follow in this video. Today I'll be discussing about that. So completing the entire syllabus is definitely important if you have enough time in your hand. Since the last 1 year or 6 months you have been studying the syllabus only, but in this remaining few days you have to only study the important points. The most important chapters or the units are your cell biology, biochemistry, plant physiology, animal physiology, cell signaling, and ecology, evolution. These two are very scoring units, very easy. The most easiest units of your entire syllabus is your ecology and evolution, so I will strongly recommend that don't skip these two units. These two units are very easy, you can easily study, so it's better not to skip the easy units. So study everything for ecology and evolution, whatever is there in the syllabus, especially habitat niche is very important. Then types of species, what is a invasive species, then what are non-native invasive, or what is a keystone species. For your evolution, geographical time scale, then types of evolution, allopatric, sympatric speciation, that is important. Then directive, stabilizing and disruptive evolution, these things are important. For your plant physiology, very important part is your phytohormones. I have made some videos of phytohormones in this channel only, you can go and check. So phytohormones are very important. Photosynthesis, you can't skip any part of photosynthesis, that is very important. Every time there is questions from photosynthesis, that is important. Phytohormones is important. Then your water transport, then your symplastic transport, apoplastic transport, that is very important. Nitrogen cycle, nitrogen fixation, that is also very important. Phytochrome, cryptochrome, phototropin, that means photoreceptors, that part. Sensory photobiology, that part is also very important. Because coming to molecular biology, you can't imagine how important molecular biology is for your SLET exam. And molecular biology, you have to go in a little bit depth. You just can't study all that you have studied in your class 12 or your graduation, post-graduation. Molecular biology holds a very important part in your SLET exam or any life science exam, but now when you are going to appear for SLET or NET, now molecular biology is little bit more advanced. You have to study it in a advanced form. You can refer to the Watson book. Now you don't have time to read the entire book, but at least you go through the replication. Don't just simply study what is replication and what are the enzymes, just go in a little bit depth. Now like in our college days, we have just studied about the helicase enzyme. It can do unwinding, but now you have to know helicase enzyme is called what in case of eukaryotes, helicase enzyme is called what in case of prokaryotes. Then you have to know about the DNA polymerase enzyme that is participating in the leading strand and lagging strand. It is different in case of eukaryotes. The leading polymerase enzyme for the leading strand is called different. For the lagging strand, it is called different. So you have to be very specific. If you don't know about topoisomerase enzyme, it is very important. You have to know about topoisomerase. Again, the name of topoisomerase is different in case of prokaryotes and eukaryotes. You have to know all these things in details. You have to know about lag operon in a very depth. So I have again added a video on lag operon. You have to know about tryptophan operon. For your transcription and translation, again you have to know in depth the name of different types of enzymes. Then again, transcription in prokaryotes is different, eukaryotes is different. You have to know about polyadenylation, splicing, 5' capping. You have to know all the names of enzymes in very depth. Additionally, you have to know one very important thing, types of different types of RNA polymerase. RNA polymerase 1 helps in formation of which RNA, 2, 3. Okay, so in case of eukaryotic transcription, 3 different types of RNA polymerases are there. So you have to know about gene silencing, and you have to know about miRNA and siRNA also, which can be little bit new for you. Coming to biochemistry, you have to study everything about biochemistry, especially the Ramachandran plot, protein, different structures of proteins, amino acids and glycolysis. You can't skip, you just can't simply read glycolysis. You have to know about each and every enzymes participating in different stages of glycolysis, and also you have to know about the net gain of ATP, where ATP is used, where ATP is released and all. Then you have to know about enzyme kinetics. If you want to skip thermodynamics, then also it's fine, but you have to study other things of the biochemistry. If you find thermodynamics very tough, then you can skip, but at least remember what is entropy, enthalpy and all. Then coming to your animal physiology, everything about animal physiology is important for your SLET exam, like your digestive system, circulatory system, especially your excretory system is important. The most important thing of animal physiology is your endocrinology. Then coming to cell biology is a very important unit of your SLET exam, and it is a kind of easy also. Okay, cell biology, you have to know about membrane transport, trafficking, okay, protein trafficking. You have to know about integral and peripheral membrane proteins. That is very important. For example, spectin and chirin belongs to peripheral membrane protein. You have to know about different signal sequence. Okay, if you have studied all these things, then only you can understand what I am speaking. If you have not studied anything, that you will not understand whatever I am speaking. I hope you have done your preparation very well, so you will definitely understand. Your signal sequence is very important. Which organelle follow signal based trafficking, which organelle follow vesicle based trafficking, that things you have to study. Then you have to study post translation and co-translation, which organelle perform this and that. Okay, that part is very important. Then you have to study little bit about endocytosis. At least you have to study which proteins, okay, for example, clathrin coated, COP1 coated, COP2 coated vesicles will target to which organelle, like that. COP1 performs retrograde transport, like that. Okay, you have to know the coating proteins, that you have to know clathrin, caveoli, all these things you have to know. You have to know about different types of lipids, okay, present in the plasma membrane. Phospholipid, glycolipid, again different types of phospholipids are there. Then coming to cell signaling, you have to know about GPCR, okay, G protein coupled receptor, that you have to know. You have to know about enzyme linked receptor. Don't forget to study about the RAS MAP kinase, that is very important, and study different types of RAS proteins. There are different types of RAS protein. For example, RAN belonging to the RAS protein helps in nuclear trafficking, like that. You have to study different functions of different RAS protein. You have to know what is RAS, okay, these are GTPase protein, like that. So RAS MAP kinase is important. PI3 kinase, if you want you can study, but you can, yes, PI3 kinase, if you find it very difficult you can skip, but RAS MAP, GPCR, that you have to study. JAK-STAT pathway you have to study. JAK-STAT is a little bit different. Why? Because the enzyme, because the receptor itself does not have that enzymatic property. It depends upon another protein, so that is important. Exceptions are important. You have to know about cancer biology. You have to know about apoptosis. Which of the following is an apoptotic protein, which of the following is a non-apoptotic protein, that you have to know. You have to know gain of function of which protein can cause cancer, loss of function of which protein will lead to cancer. Okay, proto-onco genes, tumor suppressor genes. Then coming to developmental biology, that also very important part. You have to know about WNT signaling, Hedgehog signaling. You have to know what is stem cell, cleavage of different organisms, how the eggs or the zygote gets cleaved. Then you have to know, there is a very important table which I'll try to attach here. You have to know about the different types of eggs, okay, of different organisms, and what type of cleavage they used to follow. I will attach the picture here in this video. Then you have to know about extra embryonic organs, like yolk sac helps in what, allantois, chorion, tetrapod, limb development, frog development, Drosophila, that is very important. Different genes participating in the formation of Drosophila body, that is important. Maternal and all, you can't skip that. Then your C elegance is very important. Lean 3 helps in what? Lean 39 helps in what? How many ppcs are there? All these things are important for C elegance. There are many videos in YouTube that can help you to gain marks in paper 1. For example, you search in YouTube about higher education for UGC NET. Then only you will get many information which you can use in your SLET also. So you study everything about higher education, different schemes of the government for educational aspect. Then you study about research aptitude. Okay, you just type in YouTube, you will get many videos, many playlists you will get which you can help you. So you study about research aptitude, okay, types of research, and study about different universities and their established year. You can study about different newspapers while attempting your exam. I think you already know there will be two papers, paper 1 and paper 2. Paper 2 will be your subject based, because I am a life science educator, so I am guiding you for your life science exam. But for paper 1 will be common for all, I think you know that. So there will be 2 marks for each question. There is no negative marks, so try to attempt all the questions. First you go for the data interpretation for paper 1. First you go for the data interpretation questions, because those are the easiest one. You can just look at the graph or the data given and you can just find the correct answer. A little bit maths is important, like you have to know to find out percentage, average, at least. Then SI, CP and SP, CP, all those things you have to know. At least you have to know to find out the percentage and average. Then if you are good in English, you go to the comprehension part, because there that is also similar like data interpretation. All the data will be there, you have to just answer. Then you go to the blood relation. If you are good in that, you can solve the reasoning questions. And at last you go for the GK part if you are not sure about it. Then you go to the paper 2. There will be a little bit break, you can relax, you can refresh in that break, and then you will go for the paper 2. So for the paper 2, again you try to solve the long questions first, or if you are comfortable with the short question, then also it's fine. So you just go through the first, you solve the match the following, because those will be easier. The match the following, if you know 1 or 2 or 3, then you can easily solve the entire thing. Then you go to the true and false questions. True and false are also easier, because if you know at least 1 or 2, you can solve the entire question. Then reason, assertion region, if you find it a little bit difficult, you can keep it for the last. You can solve the small questions again. So assertion region, while attempting the assertion region questions, if you find it difficult, you do one thing. First you go to the assertion, then add a because word in between, then you go to the reason. Okay, this thing is happening because of this thing. So you first read the assertion, then put a because, then go to the reason. If it seems proper, if the sentence is looking great, or if you can relate to that sentence, then it will be easy. Then the option will be the reason is certainly satisfying the assertion. I hope you can understand what I am speaking. So anyway, these are some important points that can help you in these last days. So if you want to know more, or if you want to take detailed coaching, or if you want to know some more about the exam, you can comment down below. I will be always available. So I wish you all the best for your exam, and study hard, and I hope you qualify this time. I wish you all the best, and see you in my next video. Bye, take care.